Welcome to another video. We are now going to start working on dihybrid crosses. So for this, it's going to look a lot like monohybrid crosses, but now we're going to be looking at two traits rather than one, because di means two, the prefix di. So we need to be very careful, especially when we're setting up the parents, that we keep these traits separate. So I'm going to read through the problem first, mark anything I need to, and just like I did before, I'm going to do symbols, parents, and then I'm going to transition into a new step, gametes. We did not have gametes with the monohybrid crosses. Gametes, Punnett square, and then answer any questions. So again, we're going to start very slow, make sure we understand the parents and the gametes before we move forward. So we have a heterozygous tall, heterozygous brown colored seed coat. The seed coat is what surrounds the membrane that surrounds the seed. And here's my cross. And then our other parent is a homozygous recessive short, homozygous recessive white. And it tells me to use T for height and B for color. So I have two different traits, two different letters. For each trait, I will have a capital and lowercase. Okay, so let's start with height. So I've got tall and I've got short. And I can see here I've got heterozygous tall, but here's my clue. I have heterozygous short. So short is my recessive, lowercase, and capital. My second trait is going to be color. In this case, I've got brown and I've got white. So brown and white. And it tells me that white is recessive, so lowercase b and a capital B. So that's my symbols. Okay. Now I need to write my parents. So I'm going to start heterozygous tall. So my first trait. So big T, little t. The second trait of my first parent is heterozygous brown. So now I'm going to do big B, little b. And I just list it right after one another. Cross, homozygous recessive short. So my height, so the only way it can be short would be two little t's. Keeping my first trait listed first. If you start mixing them up, it's going to get a little confusing. So just keep everything in the same order. And then homozygous recessive white. So two lowercase b's. So there's my symbols and my parents. For number 2, purebred dominant round, purebred recessive brown seed coat, crossed, heterozygous round, purebred dominant black colored seed coat. I've got shape and I've got color, and it tells me to use R and B. So for my shape, I've got round, and it doesn't tell me what the other shape is. So I can do one of two things. Usually, I've been using this before, for my shape, round, usually it's wrinkled. So I could put wrinkled, but if it doesn't tell me, I can always write the word other. But I'm just going to put in parentheses that it's wrinkled. And we said here that a round is dominant, and it said to use R for shape, so round. And then my second trait for color, I've got brown and I've got black. So even though I have brown listed first in my problem, I'm going to go ahead and keep with my pattern. And I'm going to do black and then brown. And so capital B, lowercase b. It just keeps everything organized. Now for my first parent, a purebred dominant round. So purebred dominant round, so that's going to be two capital R's. And then a purebred recessive brown. So that's going to be two lowercase b's. That's my first parent. And then heterozygous round. So big R, little r. purebred dominant black, 2 capital B's. And definitely for these, I would double check your symbols, double check your parents, and then your gametes, because eventually we're going to be making a 16 Punnett square, and you'd hate to go through everything because you messed up on one letter. Okay, and number 3, homozygous dominant yellow pod, homozygous recessive brown seed coat, crossed, heterozygous yellow pod, heterozygous black seed coat, D's for pod, and B is for the color of the coat. So I've got yellow pods, but like the one problem before this, I don't know what my other color is, and I'm actually going to go ahead and write out, because I'm dealing with colors for both. I'm going to go ahead and do yellow pod and other pod. And then I'm going to do black coat and brown coat, just because colors for both. So my yellow is dominant, and it tells me use D for pods. And whatever the other color is, maybe green, that's going to be a lowercase d. And then for my coat color, it looks like my brown is recessive, so that'd be my lowercase b, and black is dominant. For my first parent, homozygous dominant yellow, so 2 capital D's. Homozygous recessive brown, 2 lowercase b's. My second parent, heterozygous yellow, so heterozygous is one of each letter, so big D, little d, and then heterozygous black, so big B, little b. And again, I'm just being consistent, so I listed my pods or my D's first, and then my coat color second, or my B's are second. So that's how you set up symbols and parents. Now let's go ahead and move into, and let's start making gametes. Because when we did monohybrid crosses, we had one allele that we only had to worry about. So now that we have 2 traits, this would be like this law of segregation, Mendel's law, to where the gametes would segregate out, and you would have possibly 2 different alleles in each, either sperm or egg cell. And we have to look at all the possible combination, because remember, Punnett squares is the probability or the possibility of these offspring. Okay. The easiest way to do this is using the FOIL method. Okay? And we're going to look at the first pair, then the outer, then the inner, and the last. So what does that mean? So you can just kind of review this algebraic expression up here if you would like to. But to apply it to gametes, so let me take my first question here, and I'm going to write it large and then I'll redo it. Okay, so D's, that's my first trait, and here's my second trait, just like we were doing before with our parents. So if I'm doing the first pair, so first, and if I just kind of draw an imaginary line between these 2 traits, first, third, okay, and then it's going to do the outer pair, first and last, okay, and then the inner pair and the last pair, okay, FOIL. To write it as a gamete, I would go big D, little g, there's my first one. Then my second one has to be the same, big D, little g. And now I'm going to go inner, little d, little g, and then my last pair. Okay? And what would be good and makes sense is if you draw a circle around each one to represent that these are gametes. So maybe that's from the male. Okay, let's go ahead and do the other parent. So now I've got this. So first, last, big D, big G, then the outside, and these just all happen to be the same. And I'm going to draw circles around them so I know they're individual gametes. And that will make more sense when I show you how we're going to do it in the Punnett square. Okay, now for this one, we're going to go first and third, or however you want to talk about the first pair. So big B, big F, and then the first and the last, big B, little f. And then, now remember we've always said capital letter first on everything. In this instance, we want to go ahead and write it in the order that's presented to us. So we've been using B's first, so let's continue. We're going to ignore the rule of capital letters first. So little b, capital F, and then the last pair is a little b and a little f. Okay, and then just like above, these will all be the same. Okay. Okay, and then one more before we do a Punnett square. So now we've got big A, little b, big A, little b, little a, little b, little a, little b, and then next, little a, big B, ignore that rule, little a, little b, and then for the last set, okay. So hopefully that makes sense, using the FOIL method to determine the gametes. So these are the possible egg-sperm combination through the law of segregation. Now to apply that to the Punnett square, so I went ahead and drew out the Punnett square. So for me it was a backwards L, 4 horizontal and 4 vertical lines. As long as you have 16 squares in the middle. So just like we did before, in each box we put an allele for a monohybrid cross. But now we're going to put each gamete in each box. And that's why I put circles around them, rather than just making a whole string of letters. So I'm going to put, and like I've done before, I'm going to put the first parent on top and the second parent on the side. And each circle is going to go into an open box. So my A, B will go here. I'm not going to worry about a circle. And then any order on this, A, B, and then capital A, B, capital A, lowercase b. And then I'll do the same thing on the side. These are all the same. And that's why I put circles around them, just so it's easier. Now what we're going to do now is we need to recombine, go back to our rule of the dominant letter first to help us when we get to genophenotype. And so I'm going to recombine each, and I'm going to be consistent all the way throughout. So let me do my A's first. So capital A, lowercase a. Then my B's, capital B, lowercase b. And I'm going to go across capital A, lowercase a, and then 2 lowercase b. I've got capital A, lowercase a, capital B, lowercase b. Capital A, lowercase a, and 2 lowercase b. And then if I continue, I will notice I have A, B, capital A, and lowercase b. And I'll notice that as I'm going along, you notice how these are all the same? And that's going to happen sometimes. So I can either fill it all out, or as long as you remember to include it into any of your counting, you can draw a line, arrow, down showing that it's the same. Okay? But like I said, if you do geno and phenotype, do not forget to include to add all of these squares when you're doing your ratios or percentages. So if you do have any questions, just make sure you ask, and I will see you next time. We're going to expand on this and start looking at geno and phenotypic ratios. Have a good day.